Our next lecture is on movement disorders. This is lecture 27, and we're going to be talking about Parkinson's disorder as a Huntington's disorder. And these two disorders are the major movement disorders, and it's very interesting. They have some common features that sometimes makes diagnosis a little bit difficult because they are so similar to one another in terms of their symptoms. So let's take a look at these two disorders, Parkinson's disease and Huntington's disease. Again, these are the two major movement disorders that behavioral neuroscientists have studied. One of the things I like to do is to try to put a face on this. It is a disorder that certainly can strike anyone. Here's two individuals, Muhammad Ali on the left and Michael J. Fox on the right. Two, famous, one case an athlete, the other case a very famous actor, that have been afflicted with Parkinson's disorder. And these two individuals actually have done a great deal in terms of trying to raise awareness about Parkinson's, and have been very active in terms of trying to raise money for the support of research on Parkinson's disease. So when we take a look at Parkinson's and we take a look at the major identifying symptoms, this is a disorder that occurs in about 1 out of 100 individuals over the age of 65. So there's a lot of individuals that suffer from this particular disorder. So it's characterized by muscle tremors. You may be familiar with what is called Parkinsonian tremors. Rigidity, oftentimes slow movements, a lot of difficulty in terms of initiating physical and mental activity. The ability to initiate spontaneous movements, for an individual, trying to walk, for example, becomes very difficult unless there's somebody right there assisting them in terms of pointing out exactly where they should be trying to walk. These are, symptoms of this disorder that really have been noted for a very long time. It's the behavioral symptoms of depression and memory and reasoning difficulties that I think have kind of lagged behind in terms of research and in terms of our understanding of this disorder. So overall, then there's an inability in terms of maintaining one's balance. We also oftentimes have these slower than usual movements and this stiffness and rigidity, again, very characteristic of this particular disorder. In the area of the brain that we focused in on in the biochemistry of this disorder very much is involved with dopamine within the brain. So when we take a look at the biology of Parkinson's disease, here's a normal brain that you see here. Let me identify some important areas of the brain. Again, here's the two hemispheres of the brain that we see here. This is the substantia nigra here. Here's the thalamus. Here is the putamen and the globus pallidus, and of course the cerebral cortex, and here's the amygdala. It's caused by this progressive death of neurons that ordinarily are involved in dopamine. The substantia nigra is sending dopamine releasing axons to the caudate nucleus and to the putamen. And again, it's a loss of dopamine that leads to a lower than usual stimulation of the motor cortex and the slower onset of movements. So if you take a look at this figure again right here, here's our connections to the putamen area. Again, here's our substantia nigra. And again, you're getting deficits in terms of the stimulation of the globus pallidus and ultimately the cerebral cortex of the brain. So this is responsible in part for this particular disorder. The genetics of Parkinson's disease, one of the things that we know is that early onset Parkinson's does seem to have a genetic link. If you take a look at some of the concordant studies that have been done, you know, for example, if one monozygotic twin gets Parkinson's disease, what's the likelihood that, you know, gets it before age 50? What's the likelihood that the other one too, and that does too? This is, you know, highly related to early onset Parkinson's. And again, take a look at, here's dizygotic twins getting it before age 50. You know, the other still only has a moderate probability that they're going to also be diagnosed with this disorder. If a monozygotic twin gets it after age 50, again, moderate probability that the co-twin, co-identical twin will be diagnosed with it. And with dizygotic twins after age 50, again, only moderate probability. So it's only the early onset Parkinson's that has this genetic link. There's been increasing attention being paid to exposure to toxins. You know, one of the many environmental, potential environmental influences with this disorder. A pharmaceutical called MPTP, methylphenol tetrahydropyridine. It's found in herbicides and pesticides. It's also found in some illegal drugs. Ordinarily, it's converted to MPP, and that can destroy neurons that release dopamine. It can accumulate in the brain, accumulate in these neurons that ordinarily contain dopamine. Lifestyle factors, there's some interesting ones in terms of Parkinson's. Cigarette smoking and coffee drinking somehow seem to protect an individual from developing Parkinson's disease. There's research that suggests that marijuana usage actually increases the risk of Parkinson's disease. Damaged mitochondria cells seem to be common in most Parkinson's patients. Dopamine neurons may be a lot more vulnerable than other neurons in the brain. So again, these damaged mitochondria cells, many of them being dopamine-containing neurons, that really increases the risk for this disorder. A treatment, historically one that's been around for quite a while, is the drug L-Dopa. It's the primary treatment that we still use for Parkinson's. L-Dopa is a precursor to dopamine. It crosses the blood-brain barrier very easily. It is somewhat effective in treating the symptoms of this disorder. But for individuals that are in the late stages of Parkinson's, it really doesn't help very much at all. It can prevent the loss of neurons, those dopamine-containing neurons, but it can have a lot of very unpleasant side effects. Nausea and restlessness, sleep problems, blood pressure problems, hallucinations, delusions. So L-Dopa, again, is still used in advanced cases of Parkinson's. It won't help at all. But for those individuals that are at the beginning stages of the disorder, it can be helpful. There are other treatments for Parkinson's, like antioxidants. There are drugs that are available that can stimulate dopamine receptors, ones that can block glutamate as well. There are neurotropins. There are drugs that increase or that decrease cell death. Again, we call apoptosis. High frequency electrical stimulation of certain parts of the brain, like the globus pallidus, that can also be moderately effective. Transplantation of fetal neurons, very controversial, but one that has been used with some success. So when we take a look at these implants to treat Parkinson's disease, what we're doing is we're harvesting these cells from aborted fetuses. Patients do show, once these are implanted in the brain and begin to promote the release of dopamine within the brain, it can be helpful, especially in individuals that have only mild symptoms, but most patients don't really show a long-lasting benefit after the surgery. Another procedure has been, actually growing in tissue culture stem cells that have dopamine-containing neurons. And they can also be implanted in the brain, and they will produce dopamine within the brain, but this is not a long-term treatment. It works for a while, but falls short of producing a permanent change. Another movement disorder, a very interesting one, that has many of the same symptoms as Parkinson's, is Huntington's disease. And this is a deterioration in terms of motor control that usually begins at about 30 years of age, between 30 and 50 years of age. And there's a number of different behavioral changes that occur that are not unlike what you see in the case of Parkinson's, depression, cognitive impairments, memory. These are all very characteristic of individuals suffering from this particular disorder. The individual who has really made major advances in this area, a scientist, a psychologist by the name of Naney Wexler, has really helped us in terms of understanding the genetics of Huntington's disease. And I think there's a videotape that you should have watched on her, and so it's very good and it's very inspirational, her work. But in 1981, she studied Huntington's disease in a place in Venezuela on Lake Maracaibo. Here's Lake Maracaibo that you see here. Here's Colombia. Here's Venezuela. She and her research team wanted to find, or wanted to try to uncover the genetic basis, potential genetic basis for this particular disorder. And this part of Venezuela has probably the highest concentration of Huntington's disease patients in the world. So if we take a look at her research, one of the things that she did was to collect blood samples from as many HD, Huntington disease sufferers as they could possibly find. And again, there's a very high concentration of them in this one village in Lake Maracaibo. So those samples really played a key role in the discovery of this genetic marker. That discovery was actually made 2 years later in 1983. So now we have, by virtue of her work, the largest community pedigree. That is, we can search back for a long ways this particular disorder in this community in the Lake Maracaibo area. So here's what we know about the inheritance pattern of Huntington's disease. It's controlled by an autosomal dominant gene on chromosome 4. And you can see this inheritance pattern that you see here. Here you have the father who is affected, the mother who is not. And you take a look at the inheritance pattern here, is that the possibility is to have two normal offspring and then two affected offspring. So again, it's autosomal chromosome with a defective copy of a gene. So again, through her work, through the establishment of this community pedigree, she was able to make this very important discovery on chromosome number 4. So when you take a look at the genetics of Huntington's disease, you know, briefly to understand this, you know, the sequence of bases that occurs in the gene, the CAG, cytosine, adenine, guanine, they repeat themselves, and those repetitions are very important. If it's 35 and below repetitions, then an individual is safe. 36 and above, then these are individuals that are at risk. There are more repetitions, the more repetitions, the earlier the onset of this disease. So again, Huntington, that's the mutant protein that the gene codes. And again, her work allowed us to establish what this sequence was on chromosome 4. And it's been a very, you know, important advance for trying to understand this particular disorder. What does it do in terms of what we know about the brain? You know, take a look at these slides that you see here. This picture, this is an MRI. This is a normal brain that you see here. This is a Huntington's brain, and what you can see is it looks like, you know, this area has been hollowed out. This ventricular area that you see. The light areas, these very light areas, means that there's loss of tissue. If you take a look at Huntington's disease, it's primarily the basal ganglia that's being impacted. Again, unlike what we see in the case of Parkinson's disorder. So again, these are two important movement disorders that have been studied, have a lot of the same symptoms, but as far as we know, different underlying genetic and biological factors.